I want to touch briefly on certainly the safety and efficacy advancements that you've experienced to date. Paul, I'd like to first address this question to you. What would you say are the top safety and efficacy advancements or wins you've experienced in the non-viral approaches, you've selected?
So I think the biggest one for base editing itself is the high frequency of productive edits, the way we like to describe it. So because it's so precise, every edit that you make is specifically changing one base to another, and that enables us to have a very high percentage of the cells that are have the phenotype that we want. There's no heterogeneity there, and so we get very high upregulation when we say edit a promoter region and we turn on a gene. That happens very efficiently, and from a safety perspective, we don't have an off-target profile. We don't cause DNA double-stranded breaks, which activates the DNA repair machinery, and there's a lot of indels, and that has an impact, we think, on the cell phenotype and their both their efficacy for engraftment in the case of an ex vivo therapy, and also the potential that there is some longer term safety impact downstream from doing that repair and going through that stress response pathway. So I think that sort of encompasses a summary of the advantages of base editing in that regard.
Good. Okay. So Robert, I'd like to address the next few questions and topics to you specifically, considering kind of the differences between Celularity and Beam. I think we can talk through Celularity's approaches first. So given Celularity's focus on placental-derived cells, how do you see non-viral gene delivery methods potentially enhancing your cell therapy platforms, again, as it applies to your work and Celularity's work?
Well, it should be obvious to everyone listening that our focus on the placenta as a source of cells was grounded in two fundamental phenomena. And the first is that it's a highly scalable starting material. These are highly proliferative cells. We can mass produce them, and in a culture expansion system using either bioreactors or other cultivation environments, you can do a lot with a little, and you can produce large quantities of finished product from that platform. But as we're developing these allogeneic cell therapy products, mostly for cancer, autoimmune disease, and some degenerative diseases, in order to generate allogeneic products, we need to knock out certain genes to avoid graft rejection, and then knock in genes for certain target specificity, looking for target-specific antigens. So the non-viral gene delivery tools allow us to deliver genetic material safely and effectively for therapeutic effects, and give us a degree of control of those two very, very different ultimate objectives. One is controlling safety in terms of allogeneic tolerance, as well as creating a high degree of specificity for the target, for the antigen target.
Sure. Okay. Looking ahead, how do you see non-viral gene delivery technologies evolving to meet your needs, Celularity's needs, as you develop therapies?
That's a great question. I mean, look, we think that the future of non-viral technologies is really promising, right? There's lots of great companies. Beam is an example that can present safer, more versatile alternatives for gene therapy compared to traditional viral vectors, with advantages such as reducing immunogenicity, increasing the cargo capacity, and the capability to specifically target in a precise way cell types through these innovative designs. The non-viral methods are poised to revolutionize the landscape of gene therapy in general. So in that vein, Celularity is a company to provide the delivery system, right? And in developing allogeneic cell therapy products for what we're really looking long term, which is age-related diseases, like degenerative diseases of the musculoskeletal system, the neurologic system, and so on, as well as going after cancer and immune disorders. By genetically modifying placental-derived natural killer cells or T cells, we see that the opportunities are almost limitless. One of our next breakthroughs is going to be looking at the anti-aging genetically engineered NK cell, which we want to focus on the natural behavior of NK cells to seek out and target stress antigen expressing senescent cells, and that by dialing up the efficiency and lethality of those NK cells against senescent cells, we can clean up some of that material that leads to, for example, the senescence associated secretory phenotype, which is responsible for inflammaging, the chronic turned on process of releasing cytokines that stimulate never-ending inflammation, which also promotes, in many cases, the development of malignancy. So with an increased prevalence of age-related diseases and its association with cancer and all the expensive diseases that are plaguing healthcare, we think that anti-aging genetically engineered cell therapy products, which can target those senescent cells and significantly reduce some of these age-related disorders, is going to have an economic impact. And so that means the therapy can have a broader range of ultimate costs of delivery to the patient. Our genetically modified placental cell-derived T and NK cells will ultimately, we think, be both safe, very, very specific, and cost-effective.